Hello friends, good evening and welcome to the EduSat network. Our specific topic of discussion for today is biochemistry of metabolism, and this shall be a continuation from the previous time. So this shall be part 2, and friends, today will be discussed the various metabolic pathways of food digestion in our body and the disease conditions that are related to them, and of course our subject expert shall throw more light on this and take the discussion further. I would like to introduce him. We have thus in our studio Dr. Amit Bhattacharya, Assistant Professor, Department of Zoology, Ranjas College, University of Delhi. Thank you so much, sir, for coming, and with this I also request you to begin our today's lecture.
Thank you very much, ma'am. So today's lecture is in continuation with the lecture which I presented on the 20th of April. So I basically covered upon the biochemistry of metabolism, what exactly biochemistry means and what exactly the metabolism means. So understanding of these biochemistry pathways of the metabolism is very important to know about the disease conditions and to develop the various vaccines and the drugs for those particular diseases. So in the last lecture on the 20th of April, the first part of this lecture covered upon the various physiologically important structures of the biomolecules, especially the carbohydrates, proteins and the lipids. Then subsequently I went upon the carbohydrate metabolism, where I covered upon the glycolysis pathway, pentose phosphate pathway, which synthesizes the ribose sugar, which is very important part of the nucleotide which forms the deoxyribonucleic acid, DNA or the RNA. And subsequently I went for the gluconeogenesis pathway, where in our body new molecules of glucose are synthesized from the non-carbohydrate precursors. So that's what I covered in the first part of the lectures. Today I will be covering upon the glycogen metabolism, how these storage materials are synthesized or degraded when and where necessarily required. Subsequently, I will go for the fatty acid degradation. I will touch upon the cholesterol, how cholesterol is important for our body, and how the increased level of the cholesterol causes various disease conditions like the atherosclerosis and various cardiovascular diseases. And subsequently, I will go into the protein metabolism, where I will cover upon why exactly the proteins and the amino acids are required for our body and how they are synthesized. Some of the proteins are synthesized within our body and some of them are taken up from the external food sources, and how they are degraded and traced out from our body through the urea cycle. So I will come to each of them one by one. So before going into the protein and the fatty acid metabolism, let me complete up the carbohydrate metabolism, in which the synthesis of the carbohydrate glycogen is very important. So before going into the glycogen metabolism, let's take an overview of the metabolism which we discussed the last time. So when we take up the food contains various materials like the fats, carbohydrates and proteins. So the first step of these metabolism is the digestion and the absorption which takes place in our body, where these fats are degraded into fatty acids and glycerol, while the carbohydrates are subsequently broken down into glucose, while the proteins are broken down into amino acids. Subsequently, all these particular molecules funnel down into the synthesis of acetyl-CoA, which enters the citric acid cycle. In the citric acid cycle, there is synthesis or the generation of energy which is stored in the form of NADH or FADH2. And this energy reducing equivalents then go into the respiratory chain cycle, where it goes for the synthesis of ATP molecules. So I will be covering upon the citric acid cycle as well as the respiratory chain. So before going into those particular pathways, let's discuss the metabolism of the glycogen. Glycogen is nothing but the storage polysaccharides which are present in our body. So they provide the active source or the storage material for instant energy. So whenever we suffer from some metabolic disorder or we go into the starvation period, these glycogen are the one which actively gets degraded to form glucose monomers, which are then circulated and transported to the various organs of our body where the energy is required. So if we see this slide over here, in this slide there are pink part which is stained. This pink part is nothing but the glycogen particles which are stained pink in color, and this is a tissue section from the liver. So the hepatic cells are showing the storage of these glycogen, which are nothing but the monomers of glucose which are interconnected to one another. Now, These glycogens are basically made up of glucose monomers which are attached to one another in a long linear chain with the help of alpha 1,4 linkages. Subsequently, there are branchings also which are there, and these branches are because of the binding of the addition of the glucose monomers with the existing linear chain with the help of alpha 1,6 linkages. And if we see the structure of the glycogen, we can see a tree-like structure which is there, and in the center there is a protein core which is called as the glycogenin. So the glycogen metabolism starts on this glycogenin molecule which is a protein core, and this protein core has got a primer which is attached on the top of it. So to this primer, subsequently one by one the glucose monomers comes and attaches. When this glucose chain increases to about 11 residues in long, so what happens is from one of the chain the first 6 residues gets cleaved and forms a bond or branch with the existing or the adjoining chain, forming a alpha 1,6 bonding. So there is a branching enzyme which is involved which is doing the branching part. So this is how the glucose chain is increasing with the help of the addition of new glucose monomers, and subsequently the branching is also increasing. So that's how the glycogen molecules start forming or are biosynthesized. Now, in many conditions, these glycogen molecules are actively required to get degraded or release these glucose monomers so that they can be used into the glycolysis pathway, and then they can go into the citric acid cycle for release of energy which can be used for various metabolic or cellular processes. So the next part of this glycogen metabolism is how these glycogen molecules are degraded or cleaved into smaller glucose monomers. This takes place with the help of 3 major enzymes. One is phosphorylase, the other one is glucan transferase, and the third one is debranching enzyme. So what happens is this phosphorylase, what it does is it starts cleaving up the peripheral glucose monomers which are present in the glycogen chain. When this chain gets shortened up and about 4 residues are present on each of the branches, so what happens is from one of the branch, the first 3 residues of the glucose monomers get cleaved and get attached with the existing chain. So one of the branched glucose monomer which is still existing as a branch, this gets rebranched from this glycogen chain with the help of the debranching enzyme. So by this phenomenon, what happens that the glycogen now becomes a linear chain. Again the phosphorylase comes and it starts releasing the periphery glycogen glucose molecules one after another, and the chain gets shortened up with the release of glucose monomers. So this is how the glycogens get degraded as and when required in our body. Now, one of the major pathway which was discovered is called as the citric acid cycle, which is in the center of all the metabolic pathways. So if we see the diagram over here, the picture over here, these citric acid cycle, what it takes is they take up the acetyl-CoA which can be formed from the glucose to pyruvate through the process of glycolysis. Then this pyruvate is converted to acetyl-CoA which enters the citric acid cycle. This acetyl-CoA can be also formed from the amino acids as well as the fatty acids. So both fatty acids, amino acid as well as the carbohydrate glucose can go about in the formation of this acetyl-CoA. So this acetyl-CoA then gets into the citric acid cycle, where it gets into the citric acid cycle and it's taken up by the oxaloacetate, the substrate oxaloacetate, to form a citrate, and in this process there are energy which is being released. So this energy which is released is stored in the form of NADH or FeDH2, which can further go into the third stage of this metabolism, which is the electron transport chain, where this energy which is released and stored is utilized for the synthesis of ATP molecule. So let's go to the citric acid cycle. This citric acid cycle was discovered in the 19th century by the scientist called as Hans Krebs. That's why many a times the citric acid cycle is called as the Krebs cycle. So the Krebs cycle starts with the pyruvate conversion to the acetyl-CoA with the release of carbon dioxide, and in this process the enzyme utilized is pyruvate dehydrogenase. So a 3-carbon molecule which is the pyruvates get converted to a 2-carbon molecule compound which is acetyl-CoA. Now, what exactly happens in the citric acid cycle? So, the acetyl-CoA which has been formed from the pyruvate enters the citric acid cycle, or it is taken up by the oxaloacetate to form a citrate molecule. So, this 6-carbon citrate molecule, which is formed from the 4-carbon oxaloacetate by the addition of 2-carbon acetyl-CoA, gets converted to isocitrate. From isocitrate, it gets converted to alpha-ketoglutarate by the release of energy, and this release of energy or the electrons are taken up by the NAD to form a NADH molecule, which is called as the reducing equivalents. Now, alpha-ketoglutarate further goes on to form succinyl-CoA, and in this formation, again another molecule of NADH is synthesized. From succinyl-CoA, it goes on to form succinate, and in this process a molecule of GTP is formed, which is equivalent to a molecule of ATP. Then from succinate to fumarate conversion, there is a production of FADH2 molecule, which is also a reducing equivalent which stores up the energy which is released during this conversion process. From the fumarate, the malate is formed, and at the end, malates get converted to oxaloacetate with the release of NADH. So, we can just see how much of energy is being released into the citric acid cycle by this conversion of acetyl-CoA and complete oxidation in the form of carbon dioxide, which are released at this step of isocitrate to alpha-ketoglutarate and alpha-ketoglutarate to succinyl-CoA. There are number of reducing equivalents which have been produced, such as NADH, FADH2, GTP, which is equivalent to ATP. So, we can see that a glucose molecule which first gets converted with the help of the glycolysis into pyruvate, then from pyruvate it goes to form acetyl-CoA, and from acetyl-CoA the complete oxidation takes place by the citric acid cycle, where carbon dioxide is released with the release of energy which is stored in the form of NADH, GTP and FADH2. Now, this citric acid cycle is many a times called as the amphibolic pathway. So, what exactly an amphibolic pathway is? Amphibolic pathway is nothing but a pathway which is both catabolic and anabolic in nature, which means that it can help in the formation of many of the molecule. That's why it is called as anabolic, and many a times the larger molecules gets broken down into smaller one with the release of energy. That's why it is called as catabolic. So what we just discussed is this acetyl-CoA, when it is oxidized to produce the ATP and carbon dioxide, this is releasing energy. So this TCA cycle, which is tricarboxylic acid cycle, which is also called as the citric acid cycle or Krebs cycle, is acting as a catabolic pathway, while the TCA cycle is also an anabolic pathway because it provides the synthesis of many of the biomolecule, like it provides the synthesis, it helps in the synthesis of fatty acids, cholesterol and steroids, which are very important in the biomolecular aspects and processes in our body. It helps in the synthesis of many of the amino acids such as glutamate, glutamine, aspartate, asparagines. It helps in the synthesis of the heme, which is an essential part of the hemoglobin, and at the end it is essential for formation of the intermediates of the gluconeogenesis, like the oxaloacetate, which forms the glucose monomer from the non-carbohydrate precursors. So we can see this citric acid cycle is also acting as an anabolic pathway. So it is acting as a catabolic as well as it is acting as an anabolic pathway. That's why we called it as amphibolic pathway. So if we see the next picture over here, the diagram over here, we can see the acetyl-CoA, when it enters into the citric acid cycle, it goes into the various stages where it goes to form the citrate. From citrate, alpha-ketoglutarate, succinyl and oxaloacetate, and in this process the energy is released. So what is happening, the pyruvate is getting degraded into acetyl-CoA, and acetyl-CoA is further degraded with the release of carbon dioxide. So the oxidation is taking place, so that's why we call it as a catabolic pathway. But what we see in this diagram is this citric acid cycle is also synthesizing many of the biomolecules, like it is synthesizing fatty acids and steroids. From the alpha-ketoglutarate, the glutamate amino acid is being formed, while the other amino acids are also formed apart from the purines. The succinyl-CoA is helping in the synthesis of the heme, which is an essential part of the hemoglobin, while oxaloacetate, a very important intermediate of gluconeogenesis, is going for the formation of aspartate, a amino acid, as well as in the formation of many other amino acids. So we are seeing that these intermediates of citric acid cycle are very important in formation of other biomolecules such as the fatty acids, steroids, heme, and the amino acids. So that's why it is called as the anabolic pathway also. So in total, the citric acid cycle is an amphibolic pathway. Now we come into the next part of the metabolism, the fatty acid metabolism. Before going into the fatty acid metabolism, let's recap what exactly the lipids are. The lipids are broadly divided into major classes. The first class are the fats and the oils. So these are the active energy storage which are present in the animal. So the fats are nothing but the triglycerides, where 3 fatty acid chains are attached to a glycerol backbone. Then comes the second class, which are called as the phospholipids. So the phospholipids are nothing but an addition of a phosphate group to a lipid molecule. So these phospholipids plays a very important and essential part of this plasma membrane or the cell membrane. The third category comes is the waxes, which are complex hydrocarbons and which are essential for waterproofing of the cell surface, especially in the plant cells. And the last one are the sterols and the steroids, which are nothing but they contains a steroid nucleus which having 4 rings, which are called as pantothenane and cyclopentane rings. And these steroids are very important in the synthesis of hormones such as testosterone, estradiol, and many of the adrenal cortex hormones also, and they help in the normal functioning and coordinating the various metabolic pathways in our body. So these lipids also plays a very important role physiologically in our body. Now, from where exactly we get these fats? These fats we generally get from the food sources which we eat. Many a times they are synthesized in our body also by the help of various pathways, which we will be discussing right now. The food sources which are high in fat sources are the butter, cheese, oil, milk products. So all these contains high fats, and these fats acts as a thermal insulator for our body and protects us from the cold. So these fats are nothing but called as triacylglycerols or triglyceride, and if we see the structure of the triacylglycerides, we can see there is a glycerol backbone, 3-carbon glycerol backbone, and to each of these carbon there is an addition of a acyl group or a fatty acid group. So that's why due to the presence of 3 fatty acids, it is called as the triacylglycerols. Now we come into the overview of the fatty acid, what exactly happens in the fatty acid metabolism. So the fats, which are also called as the triacylglycerides, they go into the degradation process or process by the process called as lipolysis, where the lysis occurs, where the fatty acids and the glycerols are separated. Further, these fatty acids go for the beta oxidation, where further cleavage takes place, in which these fatty acids are cleaved into 2 carbon molecules which are called as acetyl-CoA by the process of beta oxidation. This acetyl-CoA can go into the citric acid cycle and go about for the complete oxidation of this triacylglycerol, or these acetyl-CoA can go and form cholesterol also by the process of cholesterologenesis. These cholesterols are very important precursors for the synthesis of the steroids, such as the hormones which are present, the testosterone, estradiol, and the various hormones which are synthesized by the adrenal gland. So these cholesterol acts as the precursor for these steroid hormone. These fatty acids which have formed into the acetyl-CoA can also form the ketone bodies. So the ketone bodies are nothing but the fuel for the extrahepatic tissue, which we will be discussing later on. These acetyl-CoA, when and where required, can go back to form the fatty acids also by the reverse process, which is called as the lipogenesis. So the lipids are regenerated from this acetyl-CoA, and these fatty acids can further go to form the triacylglycerol by the process of esterification. So we can see in the right hand side of this image, we can see all the process are the catabolism where the breakdowns are taking place, while on the left hand side of the diagram we can see the anabolism where the synthesis of these triacylglycerides or the fatty acids are taking place. So we will be discussing about each of these steps or this pathway one by one. So before going into the metabolism pathway, let's discover what exactly the fatty acids are and how they are classified. The fatty acids are being classified into two major categories. One is called as the saturated, the other one is called as the unsaturated fatty acid. These two, saturated and unsaturated, differs in the presence of double bonds between their carbon. So we can see some of the examples of saturated fatty acid and some of the examples of unsaturated fatty acid. So over here, the saturated fatty acids are shown over here, which are lauric acid, which is C12, then there is myristic acid, which is C14, then there is palmitic acid, which is C16, then there is stearic acid, which is C18, and arachidonic acid, which is C20. And if we see the melting point of these saturated fatty acids, we can see that the melting point increases with the increase of the carbon presence in these fatty acid chains. And in the next table, if we see, we are seeing some of the unsaturated fatty acid which are having the carbon double bonds present between the two carbons. So accordingly, they are named as palmitoleic acid, oleic acid, linolenic acid, linoleic acid, arachidonic acid, and we can see as the number of the double bond and the number of carbons increases in these unsaturated fatty acid, the melting point decreases down. So these fatty acids, based upon the presence of the double bond, are being classified majorly into saturated and unsaturated fatty acids. Now, how does the synthesis of these fatty acids occurs in our body? Apart from the intake of these fatty acids from the food sources, many a times we synthesize these fatty acids within our body when required. So these fatty acids are synthesized in the humans with the help of the enzyme which is called as a multi-enzyme complex, and it is called as fatty acid synthase. So if we see, there are a number of enzymes which are attached to one another, that's why the name is coming multi-enzyme complex. And if we see over here, this enzyme exists in a dimeric form. So there are two monomers which are attached to one another, and each of the monomers are having 7 enzymes which are attached to one another along with a ACP, which is acyl carrier protein. In this acyl carrier protein, we can see a phosphopentethine acid which is having a SH group, which is a thiol group, through which it attaches with the forming fatty acid chain. And on the other end, there is a cysteine amino acid which is also having a SH group or a thiol group. So this multi-enzyme complex exists in a dimeric form, and there are 7 enzymes which are present along with the acyl carrier protein. Now we will look into these fatty acid metabolism or the biosynthesis of the fatty acid, how these 7 enzymes are utilized at different steps of these metabolic processes. In the case of the bacteria and the lower organism, it has been found that these fatty acid synthase enzymes are present in a soluble and separate or individual forms, while in the case of humans, it is present in the form of a multi-enzyme complex. So this is a major difference which has been utilized by the pharmaceutical company in finding out the drug targets in the case of the bacteria or the lower organisms which may not affect the human fatty acid synthesis pathways. Now, the fatty acid metabolism or the catabolism pathway, where the degradation of these fatty acids takes place, is called as the beta oxidation. Now, why it is called as the beta oxidation process? So over here I have taken the example of palmitic acid, which is a C16 carbon molecule compound. So if we see, the palmitic acid at one of the end is having a carboxylic group which is attached with a coenzyme A group. So the first carbon which is attached to this carboxylic group is called as alpha, while the second carbon is attached to it is called as the beta carbon. So what happens is, the cleavage takes place in one round of the cycle, which is the beta oxidation. The cleavage takes place in the alpha and the beta carbon. So the C16 carbon now gets decreased down by N-2 with the release of acetyl-CoA now becomes C14. In the second round of cycle, again a molecule of acetyl-CoA is released by the cleavage of this alpha-beta bond between this alpha-beta carbon with the release of acetyl-CoA, and this chain now becomes C12 by the release of 2 molecules of acetyl-CoA. In the next round, it becomes C10 by the release of another molecule of acetyl-CoA. Likewise, it subsequently gets decreased down and a molecule of acetyl-CoA is left. So the process takes place, this cycle takes place about 7 times with the release of, at each of the cycle there is a release of acetyl-CoA, and in the end we are left with 8 molecules of acetyl-CoA which gets into the citric acid cycle for complete oxidation and release of energy. So this is the overview of the beta oxidation of the fatty acids, how these fatty acids are degraded by the specialized hepatic cells in our body and how they are degraded to provide energy for the metabolic processes. So if we see the beta oxidation process in details, we can see that the palmitic acid at the first step go for a dehydrogenation reaction where there is a release of hydrogen molecules. So these hydrogen molecules are taken up by the reducing equivalent to form a FADH2. Subsequently, there is a hydration process which is occurring, which causes the formation of a hydroxyl group, and the compound is called as hydroxyacyl-CoA. Then there is another process of dehydrogenation which is occurring with the release of the protons, and the molecule which is formed is called as beta ketoacyl-CoA, and finally the thiolase enzymes acts upon it to release the 2 molecule acetyl-CoA from one of the carboxylic end with the release of the acetyl-CoA, and the chain becomes N-2. So we are left with C14. So we started with C16, we are left with C14 with a release of acetyl-CoA in one round of beta oxidation. Now this C14 goes for another round of beta oxidation where another acetyl-CoA is released, so it becomes C12. Likewise, it goes into 7 rounds of beta oxidation with the release of 8 molecule of acetyl-CoA. So we can just imagine a single molecule of fatty acid, how much energy it can provide, and it has been found that the palmitic acid, if we calculate the number of ATP molecules which the palmitic acid is generating, it's something around 129 molecules of ATP which is being generated from the beta oxidation of palmitic acid, the complete beta oxidation followed by the citric acid cycle of these acetyl-CoA. So we can just imagine how much large amount of energy is being released from these fatty acid molecules. Now this fatty acid beta oxidation is very interestingly used by the animals. Over here I have shown the example of bears. So we know that these bears remain in a very hibernating and dormant stage for a long period of time. So during their lifespan, for a long period of time, for several months, they remain in a hibernating stage. And during this hibernating processes, they don't eat, they don't drink, they don't urinate, defecate for months. So from where exactly they are able to maintain their body temperature, and they maintain their body temperature between 32 to 35 degrees Celsius, and how exactly the various energy requiring activities are happening? And the answer is the beta oxidation of the fatty acid. So all the fats which this bear has stored and preserved in his body, what it does is during the hibernation processes, it utilizes these fatty acids and degrades them to release the energy, and this energy is then utilized for maintaining its body temperature between 32 to 35 degrees Celsius, and this energy is subsequently utilized by the various energy requiring steps or the metabolic pathways. Now we go find out what exactly happens to this acetyl-CoA apart from getting into the citric acid cycle. This acetyl-CoA can further go on to form the ketone bodies. So what exactly are the ketone bodies? Ketone bodies are nothing but they act as the fuel for extrahepatic tissues. Many times these tissues, extrahepatic tissues such as the brain cells, the muscle cells, the liver cells, the kidney cells, they are called as the extrahepatic. Hepatic means the liver cells. So the liver is the one where these processes takes place, and the tissues which are apart from separated different from these liver cells are called as the extrahepatic tissues, such as the heart cells, brain cells, kidney cells, muscle cells. So they require active energy for their metabolic functioning. So from where they get the energy? Apart from the stored energy which they have, the energy is provided in the form of ketone bodies, and these ketone bodies are formed from the fatty acid catabolism. So this ketogenesis occurs when there is a high rate of fatty acid oxidation which takes place in the liver cells. So what happens is these acetyl-CoA goes into the formation of acetoacetate, then this acetoacetate can spontaneously form the acetone, and this acetoacetate can further go on to formation of beta-hydroxybutyrate. So acetoacetate, beta-hydroxybutyrate and the acetone are combinedly called as the ketone body. So this ketone bodies acts as the extrahepatic tissue, fuel for the extrahepatic tissue. Now, if we see the structure diagram over here, these are the hepatic cell or the liver cells where this beta oxidation is occurring and the ketogenesis is happening. So what we discussed is the fatty acids get degraded or oxidized into acetyl-CoA by the process of beta oxidation, where there is an active release of energy. This acetyl-CoA can get into the citric acid cycle or can go into the formation of ketone bodies, which are acetoacetate, beta-hydroxybutyrate or acetone. So these are then exported to the various extrahepatic tissues such as the heart, skeletal muscle, kidney and brain, which actively requires the energy with the help of the blood system, blood circulatory system. And during the starvation and diabetes mellitus, it has been found there is a overproduction of these ketone bodies. So the overproduction of the ketone bodies is very harmful for our body mechanism and functioning. So it has to be checked at the right and proper time only. So the medical attention is urgently required as and when the ketone bodies overproduction are there. So what happens is these ketone bodies, which are synthesized in the liver cells with the help of the blood vessels, they are transported to the various extrahepatic tissues like the heart, liver, kidney, muscle cells, where these ketone bodies get converted to acetyl-CoA, and this acetyl-CoA now get into the citric acid cycle with the release of energy, and this energy can be utilized for the various metabolic processes. During this transfer through the blood, when these ketone bodies are overproduced, many of the ketone bodies such as the acetoacetate start coming up in the urine also, as well as the ketone bodies such as the acetone start getting diffused into the lungs. So the overproduction of these ketone body is very harmful for our body mechanism. There are different diseases which are related to the overproduction of these ketone bodies, of which some of them are ketonemia, which is called when there is an increased level of ketone bodies in the blood. Then there is ketonuria, when there is an increased level of ketone bodies in the urine. Then there is ketosis, when there is an increased level of tissue ketone bodies in our tissues. And the major causes of these overproduction is starvation, when we starve for a longer period of time, and uncontrolled diabetes. So the treatment has to start immediately. Now there are various methods through which these ketone bodies can be checked and their presence and concentration can be mapped easily. So there are various strips which are available in the market. One of the strips which is shown over here, which is called as keto sticks, in which there are small sticks which are there, which gives a color representation of the ketone bodies. So what the patient has to do is take out this strip, put this strip into the urine sample of this patient, and after 15 to 30 seconds these strips gives us a characteristic color. So we can match this color with the color chart which is present on the box, and we can immediately say that how much amount of ketones are present within the patient, and we can say that how much difficulties or carbohydrate or fatty acid metabolism the patient is suffering from. So if there is some abnormalities of carbohydrate or fatty acid metabolism, we can easily check with the help of these keto sticks, because these metabolism pathway will result in overproduction of these ketone bodies. So these ketone bodies can be checked from the urine sample with the help of these strips, and they can be subsequently treated. Now what are the importance of the cholesterol? So the cholesterol also plays a very important role in our body mechanism. But many a times these cholesterols are related to the disease conditions. But if the cholesterol is present at a optimum and a normal level, they are very good for our body functioning. These cholesterols are synthesized from the acetate, which goes on to form isoprene, then form sequelin and lanosterol, and finally these cholesterols are formed. Now, why exactly these cholesterol are important? So the cholesterol are forming the testosterone and estradiol, which are the male and female sex hormones. They are forming many of the hormones such as the cortisone and aldosterone, which are important in the glucose metabolism. Then they are forming the vitamins, vitamin D. These cholesterol are important in the functioning and are an integral part of the cell membranes. They help in the synthesis of the bile salt. So we can just see how much important these cholesterols are. But why does this cholesterol become bad to us? So the cholesterol has been broadly classified into two major categories. One is called as the bad cholesterol and one is called as a good cholesterol. So the bad cholesterol are scientifically called as the LDL cholesterol, which means low density lipoprotein cholesterol, while the good cholesterol are the HDL cholesterol, which are high density lipoprotein. So if the cholesterol in total, the desirable limit of these cholesterol presence in our body is about 200 mg per deciliter of the blood. So if this cholesterol level increases to more than 240, that's the alarming or a high cholesterol level. Now the LDL cholesterol, which is designated as the bad cholesterol, the desirable limit is less than 130 mg per deciliter, while the high level is 160 and higher level. The HDL cholesterol, which is a good cholesterol, the desirable level is 50 and higher. Because it is a good cholesterol, it has to be present in a higher quantity, and the higher level is when this condition decreases or the level drops down below 40 mg per deciliter. So we can just see the cholesterol are not always bad to our health, but if they are present in an optimum condition, they are good for our body mechanism, because they are very important for the steroid synthesis, they are important for the vitamin D synthesis, they are important part of the cell membrane. So lots of activities and molecular pathways are dependent upon these cholesterol. Now the accumulation of these cholesterol, majorly the bad cholesterol or the LDL cholesterol in our body, results in a cardiovascular disease which is called as the atherosclerosis. Now what exactly happens in this atherosclerosis? Atherosclerosis, what happens is it causes the deposition of these cholesterols or these fats on the inner lining of the arterial walls. So these arteries are very important for the blood transfer in the heart system, pumping the blood to and from the heart system. So if these arteries get clogged up, what happens is the blood flow increase or decreases due to the various parts of our body or to the heart system, and will result in heart attacks, strokes to the patient. So what happens is these arteries get blocked due to presence of high level of cholesterol. So what it does is it blocks these arteries, which causes narrowing of these arteries due to the plaque presence, and due to this narrowing, lesser amount of these oxygen carrying blood can be transferred to the various parts, and the heart's pumping capacity changes, due to which the person can suffer from heart attacks, strokes, and it can be fatal also. So higher level of bad cholesterols, fats are very bad to our health condition. So if there is some high level of cholesterol, the person should immediately go to the doctor, get it checked and diagnosed, and the treatment should start immediately, so that these kind of cardiovascular diseases doesn't happen to this person. Now let's come into the last part of my presentation, which is the amino acid metabolism. Before going into the amino acid metabolism, let's discuss what exactly amino acids are and how they form the part of this protein structure. So a protein is nothing but a polypeptide which contains a number of amino acids which are interlinked to one another. Now these amino acids are attached to one another with the help of the peptide bond. So over here a generalized structure of amino acid is shown. So we can see there is a carbon, there is an amino group, there is a carboxylic group, and another is a R group, which is a residual group which is there. So these proteins, when we take these proteins through the food sources, what happens is they get into our body, and there are various proteases, enzymes which are present within our body. So what it does is it digests them into smaller amino acids or separate them into amino acids. So this is the first part of the protein metabolism or the catabolism, where the digestion and takes place, where these polypeptide chains are cleaved or digested into smaller amino acids. Now there are 20 standard common amino acids which are present in the amino acid pool, and if we see, all these amino acids have been classified into various presence of various R groups or the residual group presence. So they can be aliphatic, they can be aromatic, they can be acidic, they can be basic, they can be hydroxylic, some of them are sulfur containing, some of them are amidic also. So based on the R group presence, so they differ in the R group. Rest of the group, the amino group and the carboxylic group, and the central carbon is present in all of the 20 amino acids. They only differ in the R group. They are broadly classified into essential and non-essential amino acids. So it has been found that of the 20 standard amino acids, 9 are essential amino acids, while 11 are non-essential amino acids. So what does this essential and non-essential means? So essential means, it means that they are nutritionally essential, which means that these particular amino acids are not synthesized in a high level in our body. So they have to be taken up from the external food sources. So from the food source which we take, these amino acids contribute and plays an important role, while the nutritionally non-important amino acids are the one which are actively synthesized in our body. But even though they are nutritionally non-important, all the amino acids, the nutritionally essential and nutritionally non-essential, are biologically important for all the metabolic pathways. So they have to be there in the body, even though we have to take protein sources just to compensate the level of the essential amino acids which are not produced in a high level within our body. Now, how does these amino acids are synthesized within our body? So in our body, these amino acids are synthesized also, they are degraded also. So the first part is the synthesis of these amino acids. If we see the cycle over here, there is a glycolysis which is taking place. So the glucose is getting converted to pyruvate, and from the pyruvate it is going into the acetyl-CoA, and from the acetyl-CoA it is getting into the citric acid cycle. So if we see the picture over here, the phosphoglycerate, which is the intermediate of the glycolysis, is forming or helping in the synthesis of serine, glycine and cysteine amino acid, while the phosphoenol pyruvate is helping in the synthesis of tryptophan, tyrosine and phenylalanine, while the pyruvate, which is the end product of the glycolysis, is helping in the synthesis of alanine, leucine and valine. And at the end, the acetyl-CoA enters into the citric acid cycle. So in the citric acid cycle, the intermediates also forms a number of amino acids, such as the alpha ketoglutarate helps in the synthesis of proline, glutamate, glutamine, histidine and arginine, while the oxaloacetates helps in the synthesis of aspartate, aspargine, lysine, threonine, methionine and isoleucine. So we can see how all these 20 standard amino acids are synthesized from various intermediates of the glycolysis as well as the citric acid cycle. So that's why the glycolysis and citric acid cycle are the central part of the metabolism, and they are very critical and essential for the body functioning. Now, the amino acid catabolism takes place in 2 steps. So what happens is, the amino acids which is being broken down or digested from the protein, diet protein, these amino acids have got a basic structure of an amino group, there is a carboxylic group and there is a R group. So this amino group from this amino acids get released with the help of the transamination process, and this amino group or the NH2 group is then taken up by the glutamate, and subsequently this glutamate releases this amino group by a process of deamination, and this amino group then enters the urea cycle so that it can be released from our body through urea through the urine. and the carbon skeleton which is present, which contains the central carbon, the carboxylic group and the R group, can go and form the acetyl-CoA, which can enter into the citric acid cycle, or it can go into the formation of ketone bodies also. Now, let's take up what exactly happens to the amino group of these amino acids. So, the amino groups first get transaminated. So, this alpha amino acid, after releasing its amino groups, becomes a alpha keto acid, which is nothing but the carbon skeleton. This amino group is taken up by alpha ketoglutarate to form into glutamate. Now, this glutamate further goes into the oxidative deamination by the release of this NH3 group. So, this ammonia group, which is taken up by this glutamate from the alpha amino acid, is released, and this ammonia group then enters into the urea cycle. So, in the urea cycle, what happens is this amino group enters into the mitochondrial matrix, the liver mitochondrial matrix, where it combines with the help of a phosphate and a CO2 group to form a carboxyl phosphate. So, the carboxyl phosphate acts as the precursor for the urea cycle. Now, this carboxyl phosphates get attached to the ornithine to form citrulline. From citrulline, the arginosuccinate is formed with the help of the aspartate. From arginosuccinate, the arginine is formed by the release of fumarate, and finally the arginine goes into the formation of ornithine with the release of urea with the help of a water molecule. So, this urea which is being formed is now excreted from our body through the urine. So, we excrete out this extra urea ammonia group from our body with the help of urea through urination. So, this is how the metabolism of this amino acids takes place. Now, what exactly happens to the carbon skeleton of these amino acids? So, what we discussed is these carbon skeletons can get into the citric acid cycle, or they can form the various intermediates of the citric acid cycle. So, if we are seeing, they are forming alpha ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, all of which are the intermediates of the citric acid cycle, and they can form the glucose also. So, these amino acid which goes into the citric acid cycle and forms these intermediates are called as glucogenic, while the amino acids skeleton which goes into the formation of acetoacetate and acetyl-CoA are called as the ketogenic. The example of ketogenic amino acid is leucine and lysine. So, through this diagram we can see that how this carbon skeleton of these amino acids are also catabolized into the citric acid cycle. So, in the amino acid, first the amino groups get released with the help of the transamination, and it goes and get released through the urea cycle, while the carbon skeleton goes and forms the intermediates of the citric acid cycle, so that they can be used into the citric acid cycle and they can release the energy for the various metabolic processes. Now, with the end of this metabolism, there are synthesis of large number of reducing equivalents such as the NADH and FADH2. Now, what exactly happens to these reducing equivalents which stores the energy which is generated or synthesized during various metabolic processes? So, the NAD which takes up the energy or the protons or the electrons through the metabolic processes get reduced to form NADH, while the FAD, which is flavin adenine dinucleotide, takes up these electrons of the energy from these metabolic processes to get converted to FADH2. Now, what exactly happens to these NADH and FADH2, and how does it goes into the formation of ATP molecule? So, the answer is these molecules goes into the formation of ATP molecule with the help of a pathway which is called as oxidative phosphorylation or respiratory chain or electron transport chain. This particular pathway occurs in the mitochondria. So, we are seeing a picture diagram of a mitochondria. This mitochondria contains a outer membrane, then there is an inner membrane which is present, and in the center is the matrix. So, the inner membrane mitochondria forms lots of cristae which gets intruded into the mitochondrial matrix. Now, what exactly happens in this respiratory chain or the oxidative phosphorylation? So, what happens is in this oxidative phosphorylation, these NADH which has stored up the energy in the form of the electrons gives up these electrons to the first complex which is present in the inner mitochondrial membrane, and from this complex 1 it goes into a molecule, transfer molecule, which is Q or ubiquinone. From Q it goes into complex 3, from complex 3 it goes to cytochrome C, from cytochrome C it goes to complex 4. So this is how these transfers of electrons are taking place, and at the end what is happening is half a molecule of oxygen along with the protons is taking up this electron to form a water molecule. So what we are seeing is the energy or the electron which is stored in the reducing equivalent such as the NADH2 is taken up through these complexes, complex 1, 3 and 4, and result in the formation of water molecule. And in this process of transfer, about 10 molecules of protons which are present in the mitochondrial matrix get transferred into the intermembrane space. So the mitochondrial matrix was having the protons, but during this electron transfer, these mitochondrial matrix is releasing these protons into the intermembrane space through the various complexes. Now what happens to the FADH2 molecule? So the FADH2 molecule, which is formed during the transfer process of the succinate to fumarate process, this FADH2 molecule is formed. This gives its energy or the electrons into complex 2, which is also a integral which is a peripheral protein in the electron transport chain. So this complex 2, what it does is it transfers this electron to Q, which is ubiquinone. From Q it goes into the complex 3, from complex 3 it goes into cytochrome C, from cytochrome C it goes into the complex 4, and finally half a molecule of oxygen, 2 proton molecules takes up this electron to form a water molecule. So FADH2 is also transferring its energy or the electrons into this electron transport chain with the release or the formation of water molecule, and in this process 6 proton molecules moves from the mitochondrial matrix into the intermembrane space. So what we saw that the NADH is helping in the transfer of 10 protons from the mitochondrial matrix into the intermembrane space, while the FADH2, which is transferring its electron from complex 2, complex 3 and complex 4, is transferring 6 molecules of proton from the mitochondrial matrix into the intermembrane space. Now what exactly is happening due to this transfer? So what is happening is due to this transfer there is a change in the electron gradient or the charge density across the membrane. The mitochondrial membrane was earlier positively charged because high presence of protons was there, while the intermembrane space was negatively charged. But as this transfer of these protons takes place, what happens is these protons goes into the intermembrane space, and what it does is it makes the intermembrane space much more positive than the mitochondrial matrix. So there is a difference of the charge density which happens during this transfer. So due to this transfer of these proton molecules from the mitochondrial matrix into the intermembrane space, there is a chemical potential and electrical potential charge density which is being created. So this chemical potential and the electrical potential energy, what it is doing is it is trying to move back these protons which has gone into the intermembrane space into the mitochondrial matrix. So it is forcing these protons which have moved into the intermembrane space to go back to the mitochondrial matrix, and this force is called as the proton motive force. As the name is suggesting, the protons are moved back with a force. So these potential changes in the potential and the chemical energy is causing this proton shift. So these protons, what happens is these protons can now come back, they can come back through the ATP synthase enzyme. So the protons which have moved from the mitochondrial matrix into the intermembrane space through the electron transport chain complexes, what happens is the positively charged develops in the mitochondria intermembrane space. So there is a high H plus concentration which occurs in the intermembrane space, and the proton motive force which has been created now helps and pushes these protons to get back into the ATP synthase through the ATP synthase molecule back into the mitochondrial matrix. So this ATP synthase molecule plays a very important role in transportation or translocating back these protons through it. So during this transfer of these protons from the ATP from these intermembrane space into the mitochondrial matrix, what is happening is there is a release of energy, and this release of energy is taken up by ADP molecule, which is adenosine dinucleotide phosphate, to attach a phosphate molecule to it. So we know that the ATP, when it combines with a phosphate molecule, goes to the formation of ATP molecule. So the ATP molecule is conserving this energy which has been released in a form of phosphodiester bond by addition of a phosphate group to the ADP molecule, and the ATPs are synthesized. And it has been found that the NADH molecule, when it transfers its electrons into the electron transport chain, there are about 3 molecules of ATP which are being synthesized, while in the case of FADH2, 2 molecules of ATPs are synthesized. So this is what is happening for the complete oxidation process, how it started from the digestion and absorption and how it completed into the complete oxidation process. So this is the basic ATP synthase molecule structure, which contains a FO and F1 particle. The FO particle is called as fragment oligomycin sensitive because it is sensitive to oligomycin antibacteria, while the F1 particle is present within the mitochondrial matrix. So the protons which is present in the intermembrane space are coming back into the mitochondrial matrix with the help of these FOF1 particles. Now if we see the ATP production which is taking place through the glycolysis, oxidative decarboxylation and Krebs cycle, we can see that from a single molecule of glucose, 36 to 38 molecules of ATPs are being generated. So we can just imagine how much of energy a single molecule of glucose is contributing. So if we see the schematic diagram over here, we can see at various points how the various different reducing equivalents are being produced, and how these reducing equivalents such as the NADH and FADH2 are going into the respiratory chain or the electron transport chain for the synthesis of ATP molecule. So in total we can see that the glycolysis or the fatty acids or the amino acids which go into the complete oxidation, the main motive of this is to release the energy which is stored in the form of ATP, which can be used for the various cellular and metabolic pathways within our body. Now the understanding these metabolic pathways are very important because these are related to various disease conditions like the inflammation, osteoporosis, oxidative stress, obesity, glucose intolerance, diabetes, low HDL cholesterol, hypertension. So understanding these metabolic syndromes is very important for creating the biomarkers, means how can we find whether the patient is suffering from these particular diseases, and the drugs. So a branch of science have come up which is called as metabolomics, which deals with understanding all these metabolites which are functioning within our body, and it helps us to understand the various genetic factors which are contributing and changing these things. And with this part I end my lecture, and in the year 2007 the whole human metabolites have been mapped up, and in the first draft about 2500 metabolites have been mapped and 1200 drugs targets have been found, of which 3500 food components which affect the various metabolic pathways have been found. So understanding these metabolites is very important as it helps in the development of the drugs, developing the biomarkers, and identify the novel drug targets which can affect and radically cure the person from that particular disease condition. With this note, I end my lecture on this biochemistry of metabolism. Thank you very much. Thank you very much.
Thank you so much, sir, for this very interesting lecture, and we've got to learn a great deal about the designated topic. So with this little time left, I would like to sign out from the lecture from both of our side, but of course we would like to thank you sincerely, sir. Thank you for coming and sharing your thoughts and ideas with us, and also talking to our students. We also request you to be present with us on other future occasions as well.
Thank you, ma'am. Thank you.
And with this, we take your leave. Thank you, friends, for watching. Have a great day.